Hello, welcome to our today's program. Today we are going to discuss chemistry of DNA synthesis, general principles, bidirectional replication, semi-conservative, semi-discontinuous and RNA priming. Now let's discuss the main objectives of today's lecture. To know the chemistry involved in the synthesis of DNA, to get acquainted about bidirectional, semi-conservative and semi-discontinuous replication, to know why RNA primers are needed for DNA replication. Now let's discuss DNA replication. DNA replication is the process of producing two identical replicas from one original DNA molecule. This biological process occurs in all living organisms and is the basis for biological inheritance. DNA is made up of two strands, and each strand of the original DNA molecule serves as template for the production of the complementary strand, a process referred to as semi-conservative replication. Cellular proofreading and error checking mechanisms ensure near perfect fidelity for DNA replication. In a cell, DNA replication begins at specific locations or origin of replications in the genome. Unwinding of DNA at the origin and synthesis of new strands results in the replication forks growing bidirectional from the origin. A number of proteins are associated with the replication fork, which helps in terms of initiation and continuation of DNA synthesis. Most prominently, DNA polymerase synthesizes the new DNA by adding complementary nucleotides to the template strand. Now, DNA structures. DNA usually exists as double-stranded structure with both strands coiled together to form the characteristic double helix. Each single strand of DNA is a chain of 4 types of nucleotides. Nucleotides in DNA contain a deoxyribose sugar, a phosphate and a nucleobase. The 4 types of nucleotide correspond to the 4 nucleobases adenine, cytosine, guanine and thymine, commonly abbreviated as A, C, G and T respectively. Adenine and guanine are purine bases, while cytosine and thymine are pyrimidines. These nucleotides form phosphodiester bonds, creating the phosphate deoxyribose backbone of the DNA double helix with the nucleobases pointing inward. Nucleotide bases are matched between strands through hydrogen bonds to form base pairs. Adenine pairs with thymine with 2 hydrogen bonds, and guanine pairs with cytosine, which is stronger with 3 hydrogen bonds. DNA strands have a directionality, and the different ends of a single strand are called the 3' or prime end and the 5' or 5' end. By convention, if the base sequence of a single strand of DNA is given, the left end of the sequence is the 5' end, while the right end of the sequence is the 3' end. The strands of the double helix are antiparallel, with one being 5' to 3' and the opposite strand 3' to 5'. These terms refer to the carbon atom in the deoxyribose to which the next phosphate in the chain attaches. Directionality has consequences in DNA synthesis because DNA polymerase can synthesize DNA in only one direction by adding nucleotides to the 3' end of the DNA strand. Now let's discuss chemistry of DNA synthesis. Oligonucleotide synthesis is carried out by a stepwise addition of nucleotide residues to the 5' terminus of growing chain until the desired sequence is assembled. Each addition is referred to as synthetic cycle and consists of 4 chemical steps. Step 1, deblocking. Step 2, coupling. Step 3, capping. Step 4, oxidation. Now let's discuss these steps. Step 1, deblocking, which is also known as denitrilation. The DMT, dimethoxytrityl group, is removed with a solution of an acid such as 2% trichloroacetic acid or TCA or 3% dichloroacetic acid or DCA in an inert solvent, dichloromethane or toluene. The orange-colored DMT cation formed is washed out. This step results in solid support-bound oligonucleotide precursor bearing a free 5' terminal hydroxyl group. Step 2, coupling. A 0.02 to 0.2 molar solution of nucleoside phosphoramidite or a mixture of several phosphoramidites in acetonitrile is activated by 0.2 to 0.7 molar solution of an acidic azole catalyst 1H-tetrazole, 2-ethylthiotetrazole, 2-benzyl thiotetrazole, 4,5-dicyanoimidazole, or a number of similar compounds. The mixing is usually very brief and occurs in fluid lines of oligonucleotide synthesizers while the components are being delivered to the reactors containing solid support. The activated phosphoramidite in 1.5 to 20-fold axis over the support bound material is then brought in contact with the starting solid support, first coupling, or a support bound oligonucleotide precursor following couplings, whose 5-hydroxy group reacts with the activated phosphoramidite moiety of the incoming nucleoside phosphoramidite to form a phosphide trimester linkage. The coupling of 2-deoxynucleotide phosphoramidites is very rapid and requires on small scale about 20 seconds for its completion. In contrast, sterically hindered 2-O-protected ribonucleoside phosphoramidites require 5 to 15 minutes to be coupled in high yields. The reaction is also highly sensitive to the presence of water, particularly when dilute solutions of phosphoramidites are used, and is commonly carried out in anhydrous acetonitrile. Generally, the larger the scale of the synthesis, the lower the axis and higher the concentration of the phosphoramidite is used. In contrast, the concentration of the activator is primarily determined by its solubility in acetonitrile and is irrespective of the scale of the synthesis. Upon the completion of the coupling, any unbound reagents and byproducts are removed by washing. Now, step third, capping. The capping step is performed by treating the solid support bound material with a mixture of acetic anhydride and 1-methylimidazole, or less often dimethylaminopyridine, as catalyst, and in the phosphoramidite method serves two purposes. One being, after the completion of the coupling reaction, a small percentage of the solid support bound 5-OH groups, 0.1 to 1%, remains unreacted and needs to be permanently blocked from the further chain elongation to prevent the formation of the oligonucleotides with an internal base deletion, commonly referred to as N-1 shortmers. The unreacted 5-hydroxy groups are to a large extent acetylated by the capping mixture. It also has been reported that phosphoramidites activated with the 1H-tetrazole react to a small extent with the O6 position of guanosine. Upon the oxidation with iodized water, this side product, possibly via O6N7 migration, undergoes depurination. The apurinic sites thus formed are readily cleaved in course of the final deprotection of the oligonucleotide under the basic conditions to give two shorter oligonucleotides, thus reducing the yield of the full-length product. The O6 modifications are rapidly removed by treatment with the capping reagent as long as the capping step is performed prior to oxidation with iodized water. The synthesis of oligonucleotide phosphorothioates, or OPS, does not involve the oxidation with iodized water and respectively does not suffer from the side reaction described above. On the other hand, if the capping step is performed prior to sulfurization, the solid support may contain the residual acetic anhydride and N-methylimidazole left after the capping step. The capping mixture interferes with the sulfur transfer reaction, which results in the extensive formation of the phosphate trimester internucleosidic linkage in the place of the desired PS triesters. Therefore, for the synthesis of OPS, it is advisable to conduct the sulfurization step prior to the capping step. Now moving on to the step 4, which is oxidation. The newly formed tri-coordinated phosphate triester linkage is not natural and is of the limited stability under the conditions of oligonucleotide synthesis. The treatment of the support-bound material with iodine and water is the presence of weak base, pyridine, lutein or choline, oxidizes with the phosphate triester into a tetra-coordinated phosphate triester, which is a protective precursor of the naturally occurring phosphate diester internucleosidic linkage. Oxidation may be carried out under anhydrous conditions using tertiary butyl hydroperoxide or more efficiently 1s positive 10 camphor sulfonyl oxaziridine CSO. The step of oxidation is substituted with a sulfurization step to obtain oligonucleotide phosphorothioates. Most DNA replication is bidirectional, which we will discuss now. Several possible molecular mechanisms of DNA strand growth would result in semi-conservative DNA replication. In one of the simplest possibilities, one new strand derives from one origin and the other new strand derives from another origin. Only one strand of the duplex grows at each growing point. In this mechanism, which operates in linear DNA, viruses such as adenoviruses, the ends of the DNA molecules serve as fixed sites for initiation and termination of the replication. A second possibility entails one origin and one growing fork, the point where DNA replication occurs, which moves along the DNA in one direction with both strands of DNA being copied. Certain bacterial plasmids replicate in this manner. A third possibility is that synthesis might start at a single origin and proceed in both directions so that both strands are copied at each of the two growing forks. The available evidence suggests that the third alternative is generally employed by prokaryotic and eukaryotic cells. That is, DNA replication proceeds bidirectionally from a given starting site with both strands being copied at each fork. Thus, two growing forks emerge from a single origin site. In the circular DNA molecules present in bacteria, plasmids and some viruses, one origin often suffices and the two resulting growing forks merge at the opposite side of the circle to complete the replication. However, the long linear chromosomes of eukaryotes contain multiple origins. The two growing forks from a particular origin continue to advance until they meet the advancing growing forks from neighboring origins. Each region served by one DNA origin is called a replicon. Now let's move to the evidences for the bidirectional replication. The first experimental support for bidirectional replication in eukaryotic cells was obtained by fiber autoradiography of labeled DNA molecules from cultured mammalian cells. Such studies have revealed clusters of active replicons, each of which contain two growing forks moving away from the central origin, thus providing an unambiguous evidence of bidirectional growth. Most cellular DNA and many viral DNA molecules replicate bidirectionally. Such viruses serve as excellent models for the study of cellular DNA replication. If DNA replication from the replicating eukaryotic cells is extracted and examined by electron microscope, so-called replication bubbles or eyes extending from multiple replication origins are clearly visible. Although such micrographs do not constitute conclusive evidence for unidirectional or bidirectional fork movement, electron microscope studies of the bubbles in viral DNA have provided evidence for bidirectional replication. If circular viral DNA molecules at different stages of replication are cut with a restriction endonuclease that recognizes a single site, the position as the center of the replication bubble with respect to the restriction site can be determined. The most common result from such analyses is a series of ever large bubbles whose centers map to the same site, indicating bidirectional replication of both DNA strands from that site. Thus, both fiber autoradiography and electron microscopy have indicated that bidirectional DNA replication is the general rule. Now let's move to the DNA replication being semi-conservative. The base pairing principle inherent in the Watson and Crick model suggested that two DNA strands are copied from the two old strands. Although this mechanism provides for exact copying of genetic information, it raised a new question. Is replication a conservative or semi-conservative process? In the first mechanism, the two new strands form a new duplex and the old duplex remains intact, whereas in the second mechanism, each old strand becomes paired with a new strand copied from it. The first definitive evidence supporting a semi-conservative mechanism came from a classic experiment by M. Michelson and W. F. Stahl. in which E. coli cells initially were grown in a medium containing ammonium salts prepared with heavy nitrogen 15N until the entire cellular DNA contained the isotope. The cells were then transferred to a medium containing the normal light isotope 14N, and the samples were removed periodically from the cultures. The DNA in each sample was analyzed by density gradient equilibrium centrifugation, which can separate heavy, light, and heavy-light duplexes into distinct bands. The actual banding patterns observed were consistent with the semi-conservative replication and inconsistent with the conservative replication. Subsequent experiments of a different design with the cultured plant cells demonstrated for the first time semi-conservative DNA replication in eukaryotic chromosomes. Apparently, all cellular DNA in both prokaryotic and eukaryotic cells is replicated by semi-conservative mechanism. DNA replication is semi-discontinuous. Let's have a look at that now. In a common I-form replication structure or replication bubble, both daughter DNA molecules are synthesized at a replication fork. Because the two strands of DNA are antiparallel, one new strand must be synthesized in a 5' to 3' direction in the same direction as the fork moves, whereas the other strand must be synthesized in an overall 3' to 5' direction relative to the fork movement. One could imagine that this would occur by having two types of enzymes at the replication fork, one to catalyze synthesis in a 5' to 3' direction and another to catalyze synthesis in 3' to 5' direction. Enzymes that catalyze the addition of deoxyribonucleotides to a growing chain of DNA are called DNA polymerases. Many of these have been isolated and studied, and all the DNA polymerases add a nucleotide to a growing DNA chain exclusively in 5' to 3' direction. Indeed, as will be discussed later, this polarity of synthesis is inherent in chemical mechanism of DNA polymerization. So if no 3' to 5' synthesizing activity can be found, how is the new strand oriented 3' to 5' in the direction of the replication fork synthesized? The solution to this problem is discontinuous synthesis of that strand, whereas the other strand is synthesized continuously. One of the template DNA strands is oriented 3' to 5' at the replication fork, and hence it can be copied continuously by a DNA polymerase extending the new DNA chain in a 5' to 3' direction. This new DNA chain is called the leading strand. Its orientation is 5' to 3' in the same direction as the fork movement. It is extending from the replication origin. The other template strand is oriented 5' to 3' end at the replication fork, and hence copying it will result in synthesis in a 5' to 3' direction relative to the direction of the fork movement. This new DNA chain, called the lagging strand, is synthesized discontinuously as a series of short DNA fragments. Each of these short DNA chains is synthesized in a 5' to 3' direction. These short DNA fragments, called Okazaki fragments, are subsequently joined together by DNA ligase to generate an uninterrupted strand of DNA. Because the leading strand is synthesized continuously and the lagging strand is synthesized discontinuously, the overall process is described as semi-discontinuous. Now, an important factor in the DNA replication is RNA priming. Let's discuss that. A primer is a strand of short nucleic acid sequences, generally about 10 base pairs, that serves as a starting point for DNA synthesis. It is required for DNA replication because the enzymes that catalyze this process, DNA polymerases, can only add nucleotides to an existing strand of DNA. The polymerase starts replication at the 3' end of the primer and copies the opposite strand. In most cases of the natural DNA replication, the primer for DNA synthesis and replication is a short strand of RNA known as de novo. Many of the laboratory techniques of biochemistry and molecular biology that involve DNA polymerase, such as DNA sequencing and the polymerase chain reaction (PCR), require DNA primers. These primers are usually short, chemically synthesized oligonucleotides with a length of about 20 bases. They are hybridized to a target DNA, which is then copied by the polymerase. Well, I hope you would have understood today's topic. Thank you.